Hey folks, and welcome to another edition of Zog Science. Today we're going to be taking a little bit of a deeper look at the nervous system, starting with sensation and sensory organs. So, what is sense? Sensation is basically three things. First of all, we've got a stimulus, and that stimulus is going to be detected by some sort of sensory receptor. The receptor must then translate that stimulus into a nervous signal or an action potential. And then finally, the signal must be interpreted by the central nervous system, usually the brain, but sometimes also can be the spinal cord. I got cut off a little bit. Today, we're going to be taking a look at the stimulus being detected by the sensory receptor. So there are kind of two different main categories of sensory receptors. The first one is actually a part of the afferent, which is just another word for the first neuron in a sequence. The other type is a separate cell, and then that cell will then somehow regulate the afferent neuron, usually by some sort of neurotransmitter being released and starting it off kind of like a synapse. So let's take a look at a couple of different senses. Let's begin with touch. The receptors for touch are found in the skin. You know, that's what whenever people talk about nerve endings, that's what they're referring to, are these different receptors. We've got four different types that we find in our skin. We've got heat, pain, pressure, and hair movement. So the different sensors are in different places. Your pain are up towards the top. Your heat and pressure, or kind of gentle pressure, are a little bit under, and then your strong pressure is kind of deeper. And then you've got nerves that are attached to each of your hair follicles, so you can kind of feel that. And you can notice that, right? Like, you know when you've been statically charged because you can feel your hairs standing up. Now, remember that action potentials are binary. They're either on or off. So the pattern of signals is what actually determines the sensation. And that can be either the frequency of them or, the lack of signals. Whatever that pattern is, that is how you determine the particular sensation. There are a couple different ways that this can occur. So, for example, if you've got gentle pressure, you've got fewer receptors that are being activated, so you have a lower frequency of actual action potentials being sent along the receptors, being sent along the nerves. Whereas when you have more pressure on yourself, right, you can tell that it's a different amount of pressure. How is that? Well, you've actually got more receptors that are being activated, so you've got more frequency, a higher number of signals that are being sent. You can also have the receptor itself react differently based on whether or not it is being applied a lot of pressure or maybe just less pressure. So that's touch. Let's talk about sight. It's mediated by photoreceptors, photo meaning light, receptors in specialized organs, which we call eyes. Kind of early eyes are not really called eyes, they're just called eye spots. And they're found in organisms such as planaria, which are flatworms. These are pretty primitive things. They're not terribly complex. And what happens is that if light hits the ocellus, we're just going to call it the eye spot, the brain responds by moving away from the light. So if it hits either one of them, the brain is going to move away from that light source, and we call that negative phototaxis. So phototaxis is movement in response to light. And so it's negative, so they're moving away from light. So they like to go to places that are darker, and that is a response that helps them to avoid predators because they're going to be less likely to be seen. Now, one of the things that the ocellus does is it basically only allows light from above to activate the photoreceptor, as opposed to light coming from any particular direction, which is good. And all these things do is they just recognize light or not light. They don't have anything else. They may have a little bit of gradation in terms of how much light they can respond to, but they're not going to be able to focus on anything. The next one that we have is the compound arthropod eye. It's a little less primitive, and what happens is that we've got each eye consisting of many facets. That's what each of the little kind of sets is called, each of the little mini eyes, and each of them has an individual lens and a photoreceptor. So you can see here the structure. You've got the cornea, which is like our cornea. You've got a crystalline cone. So we don't have crystalline cones. We have a liquid center to our eyes. And then the photoreceptor is down here at the bottom, and so the light comes in. And the thing is that they have many of them And so through using each of those, they're able to track different objects moving. Some of the arthropods are able to see in the UV spectrum, which is useful, particularly when pollinating flowers. Well, they're not trying to pollinate, they're trying to feed on the flowers. Basically, flowers have evolved the ability to reflect certain spectrums of the UV spectrum in order to help entice different insects to come land on them and to feast and also pollinate. All right, let's get into our eye. We are vertebrates, obviously, and it is the most complex. And part of the reason why is able to adjust for both near and distance vision. The way that it does that is by stretching the lens. The lens is able to become thicker to look at things that are nearby, and it is able to become flatter in order to look at things that are far away. And that all has to do with the focal point. And if you have taken physics or are taking physics, then you know some about this. We're not going to really get into it too heavily, so don't worry about it. Just understand that it has the ability to do focusing, whereas other eyes do not. So what happens is that the light is focused by the lens onto the retina. The retina is the back of the eye, and that is where the actual photoreceptors are located. And the retina is going to translate that light into a nervous signal, which is then sent along the optic nerve into both hemispheres of the brain. So if you have one eye open, you are still receiving information in both both hemispheres of your brain. There is in what is called the optic chiasm. What happens is that the half of the optic nerves go to one hemisphere, the other half go. Now, when we look at the retina, there are two different types of photoreceptors. We've got rods and cones. Rods are able to detect the absence or presence of wavelengths of light. So they're basically saying, is it light or is it dark? And then you've got your cones, which are able to detect different wavelengths of light, which is your various different colors. All right, so that was light. Let's talk about sound. Sound is mediated by receptors that vibrate in response to sound waves. Remember, sound is a wave, and it is a physical wave. It is not an electromagnetic wave, which is why you don't use photoreceptors for it. Insects have little membranes that are found on the inside of their forelegs, and those vibrate, and then they send signals into their little tiny brains. The mammalian ear is obviously much more complex, and what happens, basically its whole purpose is to channel sound waves to the cochlea, which is where the hair cells are going to vibrate and stimulate neurons. So sound waves come in here, they hit the tympanic membrane, that then translates the sound into the cochlea through a couple of other steps. Once it is inside the cochlea, it kind of spirals around in here, and as it goes, it is activating different nerves. So inside the cochlea, which is what we're looking at right here, you have a couple different things, but the actual duct that is important is the cochlear duct. And that is where the sound waves go into. They move the tectorial membrane, and the movement of the membrane stimulates these hair cells, and the hair cells will then activate the neurons, which are then sending a signal to the auditory nerve. You can kind of see what that looks like here. So you got the hairs when they are not moving. There's no sound. It is just kind of a steady beat of action potentials that occurs over time. However, when you start to begin to hear sound in one direction or the other, you get different variations of either more frequent action potentials or less frequent action potentials, and that then leads us into your brain, which your brain then interprets and adds meaning to. Your ears are also very important in terms of balance. I'm sure you've heard of this before. Your inner ear. So this is the cochlea here, and this is the inner ear. And inside the inner ear, you have fluid, and that fluid moves back and forth. And as that fluid moves back and forth, it stimulates hairs that are inside that then stimulate axons, which your brain interprets as direction and helps you with your balance. So people who tend to be more nauseous tend to also have a little bit more trouble with their balance because they are unable to, the reception of their hairs is not quite as good as other people. All right, so we've talked about sound, we've talked about light, we've talked about touch. Let's talk about chemicals, right? Chemoreception in animals is mediated through taste and smell, and the two are very closely related to one another, as we'll see in just a moment. Most animals sense their environment using chemoreception more so than any other sense. Most animals, again, this does not include us, we tend to use sight and sound, but most animals use chemoreception more than anything else. They don't have as sophisticated eyes or ears as we do. So let's talk about taste. Taste is mediated by the taste buds, which are found in the tongue. I'm sure all of you know this. There are several different types of taste. And what happens is that the taste buds interpret different molecules in the food as different tastes, and then they submit those signals to the brain just like anything else. So, this is a typical pathway that you might see, particularly for sugar molecules. The sugar molecules activate a sweet receptor, which then activates a G protein, which we have talked about before, right? And then that is going to hit a cascade of pathways, which is going to open up a sodium channel, which is then going to initiate an action potential. Now, smell is very similar in terms of its mechanism to taste. You've got odorants, which are chemicals, but they're gaseous chemicals that are binding to receptors that then are going to react almost directly with the brain in what is known as the olfactory bulb. So you have a portion of your brain that sits right over your sinuses, and you've got nerve endings that go straight from your sinuses into your brain. And that's why, in movies and things like that, people jab ice picks up or, through people's noses and they kill them. That's because your brain is right there. It's not way back in the back. It's also up here in the front. Now, part of the reason why taste and smell are so intertwined is that odorants are released into the nasal cavity as food is chewed. So as you're chewing your food, you've got these gases that are coming off the food, going up into your sinuses, and then attaching to those nerves and firing them off. And so a lot of the times, a lot of the complexity that we taste is actually smells that we associate because we're being stimulated by both our olfactory and our taste buds at the exact same time. So you don't differentiate between the two of them. You see them as being one. Pretty interesting. Some other senses that we humans don't have, but other animals do have. Rattlesnakes and other reptiles have infrared receptors that allow them to, interpret infrared lights and be able to track prey. Bats and other cetaceans, like dolphins, use sonar in order to help echolocate prey. We do not, but other animals have special pheromone receptors where they can detect hormones that have been released into the environment and then be able to follow those, usually to find mates. Fish use their lateral line, which is a line right along the side of them, in order to help detect electrical currents that they can then use to either find prey or, if they are prey, to escape predators. So thanks for watching. Next time we'll be getting into how the brain takes all these signals and interprets them. Thanks for watching, and I'll see you next time.